Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amirault, the marketing manager at Medicinal Genomics and proud member of the team that puts on the CannMed conference every year. Head over to CannMedEvents.com now to learn all about our CannMed 2021 event that will take place April 12th through 14th at the Pasadena Convention Center in Pasadena, California. And get your tickets today at our special early bird rate. And right now, through December 18th, you can register to win a free ticket to our CannMed Medical Practicum, which will take place on April 12th. It's all part of our CannMed Giving Back campaign, and it's very simple to enter. All you have to do is go to CannMedEvents.com, click on the CannMed Giving Back banner, fill out the form, and you'll be entered to win. And actually, if you follow us on social media, you will earn some additional entries. So please do go to CannMedEvents.com, check out the Giving Back campaign, and good luck. In other news, we've started a Facebook group. Each year, we are consistently blown away by the amazing presentations, inspiring conversations, and important connections that are made at the annual CannMed conference. So we decided to keep the conversations flowing online with our CannMed community group. The CannMed community is our virtual forum allowing you to interact directly with many of the CannMed conference speakers, past attendees, and other group members to discuss all things cannabis from bench to the bedside. Use the link in the show description to visit that group today and join. Our guest this episode is Dr. Mauro Maccarrone. Mauro is a pioneer in endocannabinoid research. In 1997, his team identified the receptor targets for the first two endocannabinoids that were discovered by Professor Raphael Mechoulam earlier in the decade, anandamide and 2-AG. Since then, the endocannabinoid system has been the focus of his research. At CannMed 2021, Mauro will present the Complex Endocannabinoid System and the challenge of developing selective drugs towards its distinct elements. Dr. Maccarrone's understanding of the endocannabinoid system is exceptional, and I really enjoyed picking his brain on some of the topics that have come up in conversations with other podcast guests. We discuss the technical challenges that prevented researchers from understanding the endocannabinoid system until the early 90s, how the discovery of the endocannabinoid system changed medical research and drug development, the difference between endocannabinoids and phytocannabinoids, the large number of enzymes involved in synthesizing and cleaving each endocannabinoid, and how blocking or enhancing those enzymes can affect the concentration of endocannabinoids in a person's body, how endocannabinoids work together to create an entourage effect, how an increased level of endocannabinoids can help you forget a painful experience like childbirth or a difficult run, and why researchers need to agree on a standardized formulation of cannabinoids to use for research. Before we get to that conversation, I'd like to thank this episode's sponsor, Cannabis Science and Technology, and their newest brand, Cannabis Patient Care. Cannabis Science and Technology focuses on educating the legal cannabis industry about the science and technology of analytical testing, quality control and assurance, extraction, cultivation, and processing/manufacturing. In October, Cannabis Science and Technology launched a new brand, Cannabis Patient Care, which is an educational research for patients, doctors, nurses, pharmacists, and caregivers on the latest research, benefits, and treatment options cannabis provides for a variety of illnesses. Go to CannabisScienceTech.com to learn more. And it wouldn't be the CannMed Coffee Talk podcast without some good coffee. And for that, we always turn to the Hemp and Coffee Exchange. If you don't know, hemp coffee is a healthy, delicious, natural product rich in trace minerals and nutrients, providing sustained energy without the crash of regular coffee. For more information, check out hempcoffeeexchange.com and use the promo code DRINKHEMP to get 10% off your purchase. Okay, And without any further ado, please enjoy my conversation with Dr. Mauro Maccarrone. Good afternoon, Mauro. Thanks so much for joining us.
Thank you for inviting me, Ben.
Absolutely. And we're very excited to have you as a presenter at CannMed 2021. I'm really interested to see what you have to present on. And for people who maybe aren't familiar with you or familiar with your work, I was hoping we could start if you would give sort of a brief bio of who you are and the research that you've done into the endocannabinoid system.
Yeah, well, I'm a biochemist and I work as a full professor at University of Rome and recently I moved to University of L'Aquila that is nearby. I was involved for almost 3 decades in lipid signaling, so how certain fatty molecules can bring information in our body, and I was there when endocannabinoids were discovered. And since then, I made of that a major subject of my research activity. So my group works actively on the endogenous components that we call endocannabinoids.
So that's fascinating. So you were actually there when the endocannabinoid system was first discovered.
Yeah, we issued our first paper in 1997, and you know that the first endocannabinoid, anandamide, was discovered by Raphael Mechoulam's group in 1992, and then the second major endocannabinoid, 2-arachidonyl glycerol, was discovered in 1995 again by Mechoulam and independently Sugiura. So, 2 years after the discovery of these compounds, we were able to show some properties of their receptor targets.
And so I know we're going to get pretty deep into the endocannabinoid system and all the different body systems and organs that it touches, but maybe it'd be good to start for, again, people who maybe aren't as familiar, if you could kind of give a broad brush summary of what is the endocannabinoid system and what function does it serve in the body.
Well, this is a system because it is composed of many molecules. Some are able to activate targets, and these are called agonists of receptors and are the true endocannabinoids. Endocannabinoids come from fatty acids that we eat and we know for a long time, like arachidonic acid. And then we have all the targets of these endocannabinoids, again, receptors on the surface of our cells, enzymes, little machines that break down or synthesize the endocannabinoids, other proteins on the membranes that can transport endocannabinoids in and out the cells. And therefore, it is quite an ensemble of molecules. We should say basically distributed everywhere in our body. You can find them in virtually in all tissues where they play different roles.
So, given that the endocannabinoid system seems to be so ubiquitous, as you mentioned, it's pretty much present in almost all the body tissues and all the body organs. Why do you think it took so long for researchers to really discover the endocannabinoid system and the role that it plays in the body?
This is a really a good question, Ben. But lipids and endocannabinoids again are lipids, are fatty molecules, are quite difficult to handle, and the research on these molecules is never easy. It is not yet possible to buy something from a company that all at once allows you to measure the amount of these components and to understand if they are present, and if they are, what they do. So it is maybe a purely technical question or scientific problem, if you want, that lipids are difficult to handle. And it was then after many years of research on proteins and then on nucleic acids, the information material of our cells, that we were able to look more carefully into lipidic substances. And therefore, in the 80s, for arachidonic acid derivatives, and then in the 90s for endocannabinoids, research focused on these molecules and was able to make a kind of quantum leap since then.
Interesting, interesting. And now, since the discovery of this system, and as we mentioned, it is involved in so many different organs and tissues, how has that knowledge sort of changed how, medical research is done into different disorders that may affect these systems?
Well, it is true that the discovery of endocannabinoids immediately attracted also pharmaceutical companies, because the endocannabinoids themselves and the targets that they hit in the cells uh, can represent novel targets for novel therapeutics in a time where we did not have so many new totally innovative targets to look at. Therefore, in a few years, basically 1.5 decade, we could say, uh, enzymes that break down endocannabinoids became targets for intense research efforts by all major pharmaceutical companies all over the world. And so were the receptors. So in a way, these compounds have filled the gap, and in this gap, therapeutic perspectives seem to be very, very strong.
Great. And as I was doing the research for this conversation, I did come across your paper entitled Endocannabinoid Signaling at the Periphery 50 Years After THC. It's quite extensive look at the endocannabinoid system. And as have mentioned several times, all the different organs and body systems that it interacts with. And I will put a link to that paper in the show notes in the description so everyone can take a look at that. I very much encourage people to read it. And a lot of heavy hitters in the co-authors of that paper, including our dear friend here at CannMed, Dr. Raphael Mechoulam. I was hoping we could kind of use maybe that paper as a framework to explore the endocannabinoid system a bit more. So maybe let's start with covering some of the different systems that the endocannabinoid system interacts with and that really show that promise for therapeutics that you mentioned.
Yeah, but first, well, thanks for the quotation. And I like to say that was a community paper that I happened to put together and coordinate with colleagues that have contributed the most to the field since the discovery of THC, tetrahydro delta 9 tetrahydrocannabinol. And this gives me the chance to make a clear point. Plant extracts, so the phytocannabinoids like THC, were discovered approximately 30 years before the endogenous cannabinoids. So we have to clearly make a distinction between the endogenous compounds that all of us we do have, and those compounds that some of us decide to take in different ways by the plants. And these are very different. So cannabinoids were discovered almost now more than 50 years ago in 64, again by Raphael Mechoulam. And only after that we understood that there are endogenous compounds that activate the targets accidentally activated also by the plant extract. And therefore, cannabinoids are something different from the endocannabinoids. The endocannabinoids and the endocannabinoid system certainly hold a lot of promise for the central nervous system, where it can contribute to mood. There are some indications whereby depression can take advantage of an inhibition of the cleavage of the endogenous cannabinoids, for instance. But also at the periphery, bone homeostasis, rather than reproductive functions, rather than energy homeostasis, which means food intake, the pleasure of eating food, the search for palatable food, and the way we take energy out of food, making more fats like lipogenesis or breaking fats in the lipolysis. All these aspects are, in a way or another, under control of the endocannabinoids.
So that's interesting when you're sort of making that distinction between the endocannabinoids and the phytocannabinoids. And I think, at least I understand that, folks who benefit from cannabinoid therapies typically may have a deficiency in their endocannabinoids that the body is creating endogenously. And from what I understand in talking with you, that the endocannabinoid system is a very complex system with a lot of different things at play. So how many different ways could someone develop a deficiency in their endogenous cannabinoids?
Oh, there are actually many, which makes the field complex but also challenging and promising. Just to give you a general guideline, each endocannabinoid is biosynthesized by one or more enzymes, and then it is cleaved by one or more additional enzymes. Uh, if you are able to block or enhance any of these enzymes, you can modify the final content of endocannabinoids, what is called in general the endocannabinoid tone. And this, in our body, apparently, the way we take advantage of endocannabinoids as very special signals is through metabolism. So there is a metabolic control. In other words, the way our body decides how much endocannabinoid must be in a certain cell at a certain point can make a really difference between health and disease.
So you mentioned that there are a lot of different endocannabinoids. Roughly, how many different endocannabinoids do we know of, and are there more?
So far, we can say that we know a dozen, of which still the very first two to be discovered, anandamide and 2-AG, those that I mentioned before were found already in 1992 and 1995. Those two remain the most interesting because apparently they are the main signals that our cells use. Yet we have to admit that of the quite many others, we don't know yet much. So maybe we have not learned enough about anandamide and 2-AG that we decided to investigate thoroughly the others. And therefore, we cannot really predict if another of these endocannabinoids can be more relevant for a certain specific condition, maybe a specific disease.
One of the topics that always comes up in talking about cannabis medicine is this whole idea of the entourage effect. And I'm curious to get your thoughts on that from an endocannabinoid perspective. And we were just talking about there are are many different endocannabinoids. So is there sort of an entourage effect that's happening endogenously as well?
This is also a very interesting question, Ben, because we have learned, and it is clear, that there are compounds that do not act directly on the receptor targets of the endocannabinoids, but can potentiate the activity of other endocannabinoids on those receptors. Why? Because they interfere with the metabolism of those endocannabinoids. For instance, by blocking the hydrolytic enzymes. And this entourage effect seems to be relevant under many, many circumstances. And again, also the endogenous compounds, not to mention the plant extracts that we might ingest for whatever reason, also the endogenous compounds can play this entourage effect.
And you mentioned earlier when we were talking about how it took so long for researchers to really fully understand the endocannabinoid system. You mentioned that it's hard to test someone for the levels of endocannabinoids that are in their system, or at least it was earlier. Is there a better system for doing that now? Can someone, go and get tested and get lab results in terms of figuring out if they have an endocannabinoid deficiency?
Basically, the techniques remain sort of highly specialized. You have to use what is called liquid chromatography mass spectrometry. Maybe it is true that now you can find these techniques much easier than it was just a few years ago. However, you need a high level of specialization and a high level of expertise to do these assays. It is not like for other substances in our blood that with a drop of blood in 20 minutes you have your data. It is still quite challenging. There are investigations looking for alternatives, and this might change the scenario in the coming years, but so far you still need an high level of specialization and quite sophisticated machinery to do these assays.
And I would imagine too, would the level of endocannabinoids in a person's system sort of depend on different stresses, or as you said, it can be part of metabolism. So do the levels sort of fluctuate throughout the day?
This cannot be really said conclusively from a scientific point of view because, to my knowledge, it has never been tested in a very solid scientific manner. But what we know is that there are changes there are during the day rather than during different conditions. For instance, the estrus cycle in women. And certainly there are changes from individual to individual, so interpersonal changes. We cannot yet assign a meaning to a certain value. We are not at the stage of defining threshold values below which everything is okay for the whole population and above which there might be a problem. We are, let's say, starting to learn how these differences could be meaningful for our health.
Yeah, that's interesting. And I mean, I'm a runner, so I am familiar that, as I believe, and you can certainly correct me if I'm wrong, that anandamide is sort of what's credited with the runner's high. Do I have that correct?
Yeah, but it depends on the way what you mean with high. What I can tell you is that
Right.
uh, there could be an increase in anandamide, but maybe to cope with the pain that can be associated with an extensive physical exercise, because endocannabinoids like anandamide are certainly analgesic. So there is the hypothesis that when you practice a lot, but also in other circumstances, like in the delivery phase, when you are delivering a baby, an increase of endocannabinoids help you to cope with pain. Uh, there is also something extra, something more that seems very interesting. The fact that an increased level of endocannabinoids might help you to forget aversive memories. So to forget something that you did not really like, but that in terms of evolution must be done again. For instance, once again in a parturition. So the pain that the mother can feel in delivery is forgotten better with an increased with an increase of anandamide. So it's not that you feel better as if you were smoking a joke, but you might feel less pain and forget something which is negative uh, due to this increased level of endocannabinoids.
So that's interesting. So you're telling me that my body is helping me forget the pain from that run so that I'll get up and do it all over again the next day.
Exactly. And you like to do it again, and you don't associate the negative memory or the pain feeling that you had.
That is certainly a clever trick because
Yeah.
'cause I do get out there and do it again. I did want to talk to you a bit more about, cannabis therapeutics, or maybe just, phytocannabinoids. And so,
Yeah.
one of the newer aspects that we've been seeing research on in at CannMed is looking into sort of these different types of cannabinoids, whether it be in acid form or even in cannabinoid varins. So I was wondering if you could speak a bit to sort of how these different forms of the cannabinoids, how does their effect differ on the body?
Well, as for the acids, you know that we also have endocannabinoids that are acidic, like N-arachidonyl glycine. It is a natural substance, an endogenous substance with an acid tail. So it is an endocannabinoid acid, actually. And we know that it mimics some of the properties of anandamide, for instance, with a with affinity for type 1 cannabinoid receptors, with an anti-inflammatory activity, and again analgesic properties. The same can be said for the main cannabinoid acid, that is a phytocannabinoid this time, a derivative of THC, an 11 oic acid, but besides the chemistry behind, is an acid form of THC. But this one is a form of excretion, is a metabolite that has to be wasted by the body. So, if you add acidic tails to endogenous or plant derived cannabinoids, you certainly have different effects. Um, Maybe what is more complex is to understand how these molecules, along with the varins, for instance, the THCV, the tetrahydrocannabivarin, uh, they play together with the other components of a plant extracts or the endocannabinoid system. And this complexity of molecules working together is a real challenge to be understood. Uh, Having THC, having CBD, cannabidiol, having both together, or having them along with other less investigated compounds can make really a huge difference in the biological activity that they have.
That's got to be the biggest challenge of all, is that there seems to be so many different compounds at play here, and it seems to be very individualistic too, in terms of what the need is in the individual body. So I imagine that must be a very big challenge for anyone who's trying to develop a therapeutic.
Yeah, and you know what I like to say as much as I can is that it is a pity that we did not yet decide which are the standardized formulations to be tested also in therapeutic terms. Because if we keep on using different formulations, of which maybe you only know the amount of THC and CBD, but you do not really, I, I don't want to seem unpolite, but you don't seem to care at all about the others, you actually miss the opportunity of learning from what you are doing, because using different things will not give you a solid knowledge that will finally help you to advance your understanding. We have to clearly say that cannabis is not one. You can have as many cannabis, as many formulations are going around, are being used, or can be used. So the composition of each formulation, and also the amount of the different components of each formulation can make really a difference. And we should now decide how to standardize these things, with the hope of learning maybe from mistakes, but in a 5 year time, 10 year time, we will really know better what is good, what is less good for our health.
That's a really interesting point, and it really touches on, a lot of the different conversations I've had on the podcast and presentations that we've had in previous CannMeds. Is first of all, from a testing perspective, it's difficult for them to have assays that are going to detect all the different cannabinoids, or and we haven't even gotten into terpenes and flavonoids and stilbenoids.
Oh
And
yeah.
I am interested to get your thoughts on that. But from a testing perspective, it's hard for them to really get a full profile of what the product is. From a cultivation side, it's very hard for them to produce a consistent product. And then it's interesting to hear you say from a research perspective that it's difficult for you to have a real standard medicine to work off of. So it seems to be a problem that really touches all the different aspects of
Yeah,
the
yeah,
industry.
yeah. Yeah, and is a problem that finally has to be faced. And I'm perfectly aware that it is not a trivial problem if you consider that we know that from a cannabis extract, from cannabis sativa or cannabis indica, both used to make formulations used in therapy, you can expect at least 110 different phytocannabinoids, and on top of them, at least 440 non-cannabinoid components like terpenoids, flavonoids, and so many others. So it is a challenge, but it is just because this challenge can lead to real good drugs, therapeutic drugs, that we have to take it in the proper way. There are no shortcuts to reach in an easy way a good drug at the end. You have to study properly the composition and the effects of these combinations. And maybe if we did that 20 years ago, now we would really know so much more, then we could have more therapeutics that are active, effective, and are based on these cannabinoids.
Yeah, it certainly would have been nice to get a jump start 20 years ago, but I know,
Hmm.
at least here in the United States, that was not possible given the laws. But thankfully, those seem to be loosening, and research has really come a long way. So we're excited in a lot of different ways. Um,
And if, oh, And if I may add, Ben, another clear distinction that we should make is between recreational cannabis and medicinal cannabis, because a therapeutic use of cannabis under the responsibility and the control of a physician that continuously looks looks after his patient is something, and to my understanding, that could be made as free as possible. On the other hand, a recreational use of cannabis can be dangerous. It's not at all true that these compounds are harmless, especially if you think of what is going on is available also through internet and other means to young people. You can really find formulations where THC itself is 10 times more concentrated than in others, and other formulations where you also have unexpected compounds. So a free recreational cannabis can be more dangerous than usual, and certainly I have the feeling that certainly this mixing up the two, recreational and therapeutic, did not help the medicinal cannabis.
Yeah, I think you're right. Um, at least for a long time here in the United States, and actually, it is still the case today. It's been classified as a Schedule One drug, which states that there is no known therapeutic use, which I think we've pretty much disproven. And certainly, your work and the work of many others have done that. So
Yeah.
hopefully, um, and sooner than later, we can remove that tag. And before I let you go, because I know I'm coming up on the end of the time that I promised to keep you, I did want to talk a bit more about terpenoids, flavonoids, stilbenoids. So sort of these compounds that are present on the plant that aren't necessarily cannabinoids. So therefore, they're not
No.
really interacting as endocannabinoids. At least I wouldn't think so. So, but are they sort of mimicking other endogenous compounds within the body to kind of help those cannabinoids do what they need to do?
Well, for some of them, there are activities known for decades, like anti-inflammatory, antioxidant. And some may also have specific targets that are certainly different from the targets of the endocannabinoids. So again, the concept of the entourage effect could come out here, in the sense that the combination of these compounds, non-phytocannabinoid compounds, together with some phytocannabinoids that are present in a formulation, can change the reaction of the endogenous system to them. And we do not know much about these interactions and these interference among components. And once again, this is why we must standardize formulations and investigate the effects in a systematic, very sound manner.
You know, some people might find it maybe discouraging to hear how little we still know or how much work we still have to do. But I find it fascinating and exciting that there's still so much more to know and so much more potential within this plant and within this system as well. So I guess with that, that's a good place to stop. First, I want to thank you for joining us here on the podcast. And before I let you go, I want to give you an opportunity if there's a website that you'd like to point people to, or if you, I don't know if you're involved on social media or anything like that, any sort of plug you want to give to the work that you're doing so people who are interested can learn more. It'd be great to share that.
Thank you so much, Ben. I suggest people to look through PubMed, which is an official site, open, public, where scientific studies are reported rather than uncontrolled or less scientific, not to say gossip, that you can find easily elsewhere. So please, when you want to know more, try to have a good source of information where it is clear that things that are claimed because there is a scientific background well recognized by the scientific community. So once again, you can go to PubMed, an NIH site, and under my name you might find papers, and whatever you want to know more, I'll be glad to keep in contact and exchange information.
That is excellent, and that is excellent advice. In things beyond cannabis too, always make sure that you're trusting your sources.
Exactly.
So Mauro,
Yeah.
thanks again for joining us, and I look forward to seeing you out in Pasadena for CannMed.
Thank you so much, Ben, for the opportunity, and I look forward to meeting you and many other colleagues and friends to discuss this fascinating topic.
I hope you enjoyed my conversation with Dr. Mauro Maccarrone. Check out the links in the show description to learn more about the topics we discussed, and thanks again to our sponsor, Cannabis Science and Technology. Some sad news to report, but this will be our last episode for the year. That's right, no new episode on December 23rd, as we take a little break to enjoy the holiday season. Don't worry, we will be back with a brand new episode January 6th. I hope you'll take this opportunity to get caught up on one or more of our past episodes. And if you have a request or suggestion for a future guest, we'd love to hear from you. Email us at info@canmedevents.com or hit us up on one of our social media properties. We're on Facebook, Twitter, Instagram, and LinkedIn. Just search for CannMed Events. And finally, if you enjoyed this episode or any of our previous shows, we'd appreciate it if you hit that subscribe button on your podcast player and gave us a 5-star review. All right, that's it. Happy holidays, Happy New Year, stay safe, stay healthy, and be sure to come back for the next episode of the CannMed Coffee Talk podcast.